So welcome everyone. Uh, it's such a blessed day that we are back to our normal routine. You can see I'm sitting in my office. On the LUMS campus also, I saw some of the students walking. Allah has some very special karmo fazal and his khas rahmat on all of us, that we are out of this crisis, which is much more deep all around the world, as you can see, which is still going on. We should not be, lowering our guards. We should be really very careful. But it's a big day. It was a strange feeling walking all the way up to the office today. To see that we are going to get back to business, our research, our teaching, our labs, and slowly we'll see, inshallah, all the students one by one on campus in a phased manner. So the course we are going to get into is BIO221. It's genetics, and we are going to cover principles of genetics, fundamentals of genetics. We'll start a bit slow, but in a next couple of lectures, we'll take off and go into, deep understanding of genetics. Also, before I get into the details of today's lecture, I would also like to welcome all the bio majors in particular. This is the first course in the sequence of bio major courses, and one of the fundamental or foundational courses in your long journey in life sciences or in the field of molecular biology. I shared with you the outline. The outline is very detailed. Yesterday, I sent you the course outline. And I would like to draw your attention to the honor code I mentioned there, in which I have talked about academic honesty, integrity, So it's a coincidence. I would say it's a simple by chance that we are in such a situation. But normally in my major courses, I never invigilate my exams. I want you to be responsible for each and every action, each and every step you take throughout this course. And I want you to realize that if you do anything wrong, it will have a long-term consequences in your lives. If you cheat today, you will grow up as a cheater whole life, and you will never be successful in life. I want you to be successful. I want each and every student of my class, of my school, to be proud of his or her achievements. And one becomes happy and enjoy achievements and success when one truly owes his and her achievements in a true sense. So I had been teaching online during COVID break, in summer, in junior courses in April, May. And those students will tell you, and if you talk to your seniors in biology department, they will tell you, Tariq is going to give you assignments. Tariq is going to give you exams, and he's going to leave the room. And he's going to trust each one of you, because getting A plus or A or B, any grade in genetics is not important. What is important is your success, and that you own everything, um, that you grow as a responsible person and show highest degree of integrity. So if you haven't read, I encourage you to go and read that outline in detail. And today we are going to start. Today we are going to start the first lecture, and let me share the. You have to excuse me because I forgot my glasses at home. Where is share screen? Yeah, here it is. So can you all see the screen? Is my screen visible?
Yes, sir.
Okay, good. Let me go on. So we are going to, throughout this course, we are going to learn about the fundamentals of genetics, and the main theme of the course is how can we use genetics to understand cellular function? How can we understand biological function? For example, we inhale air and we exhale air. Each and every body part, each and every cell in our body, it gets an access to oxygen, and then the gaseous exchange takes place. How it takes place? How can we study these functions or these questions? How, while teaching, I'm moving my hands. When we walk, our whole skeletal system, the bones, they are all attached with muscles. So how an organism is able to move muscles? So the list of questions is enormous. You come up with these questions in your daily lives, but we don't know the answers. And during this course, for example, genetics deals with genes. So how can we discover genes? This is what we are going to learn in this course. Once we discover a gene, how we are going to characterize that gene, how we are going to understand the function of that gene. So if the list of questions are unlimited, during this course, we are going to learn how to bring answer to these questions using genetics. So this is my favorite slide, in which, this is like a triangle, a simple triangle, but this is trying to explain you that, the ultimate goal of scientists is to understand the function. As a scientist, you always dream of discovering function of a cell, a tissue, an organism, and how the whole body functions, regardless of the kind of organisms you have. So I have my glasses now. There are two approaches. There are two approaches using which we can understand the cellular function. For example, so one of, So you can understand function either through genes, or you can also understand function through proteins. Historically, people, so 100 years ago, when somebody was interested in some cellular function, people were taking gene-based approach, which we refer to as genetics, or they were taking protein-based approach, which is referred to as biochemistry. Both were ultimately finding the function, okay? Because 100 years ago, or even less than 100 years ago, until 1950s or so, we were not sure that genes and proteins, they are basically connected. So people who are taking proteins as the key molecule, going through biochemical approach, they thought that, you know, it's the biochemistry or it's the proteins which are responsible for all the cellular functions. And similarly, geneticists, they were, while taking gene-based approach, also were able to characterize function. And it continued even after the discovery of structure of DNA, you know, till today. These two approaches are some of the best approaches to understand cellular function. However, these approaches are linked. With the advent of molecular biology, what we learned that genes, they encode proteins. And, you know, if you understand function through proteins, you can actually go back to genes, isolate genes, and characterize the protein. Geneticists who were taking gene-based approach, for example, in my lab, we are functional genomics lab, we are going through gene-based approaches. And when you're dealing with genetics, you are trying to understand the function by deleting genes, by playing with the, you know, specific genes, and then see if I see change in the function of the cell, tissue, and the organism. But it does not mean we don't work on proteins, because after the advent of molecular biology, so you are also learning a whole course in molecular biology by Dr. Shahzad and Afzal. So in molecular biology, we are simultaneously dealing with genes and proteins, both. But for this course, we will focus on genetics. So how a geneticist tackles a question. For example, a geneticist is going to understand wing formation. Or for example, in my lab, we work on fruit fly. Let's say we are interested in, you know, the wing development of fly. How are we going to understand wing development using genetics? So what we do as geneticists, we are going to make a mutant. We are going to create a fruit fly in which the wing is deformed or wing is missing. How do we do this? We will learn this in this course. But a geneticist will try to create an organism, a fruit fly or a butterfly in the context of this question, which is unable to fly, unable to utilize its wings, or where wings are deformed, there are some defects, but rest of the organism is normal, is viable, okay? And ideally, we are looking for, you know, single gene mutations in those organisms. Other people can ask this question: how yeast utilize sugar? And a geneticist will try to create a mutant yeast which is unable to utilize sugar, which means he will mutate, he will create changes or treat yeast with certain chemicals, create DNA-based mutations where genes are going to be defective, and otherwise yeast will be fully viable, alive, normal, but it will be unable to utilize sugar. In contrast to geneticists, same question can be addressed by biochemists. For example, a biochemist is going to collect many butterflies or many fruit flies. He is going to create an extract. He'll kill all the organisms. He'll create an extract of all the proteins in the cells. And then he's going to purify those proteins using biochemical approaches and try to find components which are having some muscular activity, or have some, which means, so protein in the ependyne or tubes, that will not show you some muscular activity. A biochemist will design some assays, let's say ATP utilization assays, or other muscle protein specific assays, and then try to see I have some proteins which may have a role in wing development. The yeast answer to sugar metabolism in the yeast can also be approached using biochemical approach. So a biochemist is going to grind the yeast. It will have all the yeast proteins in an extract and then fractionate. We call this fractionation because you have columns, and then on those columns they pass proteins, and proteins are separated on their bases or charges, different characteristics of proteins. And then biochemistry is going to see if I have some fraction of the proteins separate from rest of the cellular proteins, and these proteins can digest the sugar. So both approaches can be utilized to understand cellular function, but during this course we are going to approach the genetics, and we are going to see how we can understand cellular function using gene-based approach. So I explained you already these things. So now, what is genetics? Genetics is simple as if you pay attention to the word genetics, is actually already telling you what is genetics. It's a discipline of biology which deals with inheritance of genes, which deals with two fundamental concepts, heredity. And you study genes, inheritance of genes, and when we talk about inheritance of genes, it means from parents to offsprings. And what is conserved from parents to offsprings is, we always will have babies. Humans will always have babies, and cats will always have kittens. There will be no chimps in our family, in our species. So this preservation of ancestral characters, generation after generation, and that is called heredity, and that is what is genetics. Okay. Now, in genetics, since we play with the genes, we, for example, using genetics, you want to improve some crops, you want to understand gene functions. You won't be able to do these things if we don't have variation in the genes. And if you recall the BIO 101 lecture, we did talk about these two concepts. So variation is deviation from heredity. Deviation from heredity does not mean you will start having chimps in your family, or cats will start having, instead of kittens, will start having pups. No, variation means actually variation in some characters. And a good example will be, you just think of your own family and you will see none of the two siblings in your family look 100% alike. Some are tall, the others are fair, some may have curly hairs, the others have very silky hairs. So these traits, characters, they are actually encoded by variation. And how this variation is encoded, because there are differences at the level of genes. For example, my hair color is gray since last 30 years, but my other siblings don't have such strong appearance of gray hairs in their early age. It means the gene which encodes the pigment in our hairs, we carry the same gene, but there is some variation, some differences. The nose shape, the height, all these are encoded by collection of genes, but then there is variation, and that makes us different. And remember, variation can be of two types. It can be discrete variation, and discrete variation means black and white, okay? For example, you may think of discrete variation in terms of, the best example would be, I gave you my hair color. In plants, for example, height in plants, I'm saying, in plants, height can be a discrete variation, example of discrete variation. Flower color, red, white, these are, when you have two states, I would say binary states, as you say in computer science or in maths language. That is what discrete variation. But there can be continuous variation as well. And continuous variation can be, for example, in humans, the height is a continuous trait, continuous due to continuous variation. The yield in crops is example of continuous variation as well, due to continuous variation as well. So if we don't have variation, we cannot study genetics. Genetics, in the success of genetics is based on variation in the genes, okay? So what is basis of heredity? What is the underlying cause of variation within the species? It's basically genes. And let's learn about those genes. This is, you can see variation. Can you see on the same cob? Different colors, dark purple, pinkish, whitish, yellow, etc. So you all have learned that, we all carry DNA, okay? And genes in DNA are, I just try to draw gene here, for example. And a gene structure is there is promoter element. There is then this coding sequence, ATG, sorry, ATG, etc. And then, you have stop codons, you have the transcription termination site, poly A site, and it gets transcribed. And after transcription, it gets translated into a protein. And protein is the functional molecule of a gene. Protein is the one which goes within our cells on specific position within the cell and performs a specific function. But when we say the fundamental unit of heredity is gene, we are also saying the cause of variation is also genes. So in normal case, you have in a single cell, for example, in a single eukaryotic cell, you have, cytoplasm, you have the nucleus, and here you have the nuclear genome. In humans, we have 46 chromosomes. All these 46 chromosomes, they contain nearly 20,000 genes in such forms. So these 46 chromosomes, they have these genes. Now, as I said, not every individual within the same family contains same 100% identical gene. 99.9% genes are identical. 46 chromosomes are identical. DNA sequence on those chromosomes are identical. But there is variation. And that is very useful for us as biologists. So, any gene can exist in multiple forms. For example, I gave you example of, hair color, white and black. Let's say my brother has this gene and my cells, let me draw my weird cell. I also have the same 46 chromosomes. I also have the same gene, but there is some variation in this sequence. When my gene is getting transcribed and translated, protein is exactly similar, not identical, is exactly similar to the protein in my brother's cells, but my protein contains some variations at amino acid level. Okay, and so it makes my gene different than my brother's gene. It's the same gene, but not identical gene. So these alternate forms of genes are called alleles. My gene is also is in my hair cells, but it's not able to contribute to the black pigment. My brother's gene is able to contribute to black pigment. So this way, you can have not just two forms, you may have multiple alternate forms. And these different forms of genes, they are referred to as alleles. Okay, and we designate them differently. For example, in my brother's genome or cell, we will call this since it's white, it's black. In my case, we will call this like this. If somebody is going to have, a different, same gene, but a different change or mutation, we may have it like this. Okay. So there are different forms. So here, this example is there. Let's say this is the normal gene, which gives you black. Let's say this is alternate form. This is the allele B prime. This is now B2 prime, B3 primes, and so on. So it's in genetics, it's your call. You will be able to decide, I'm going to give it name B, A, X, Y, whatever you want. Now, this DNA level difference, which we have named alleles, we call it allelic variation. This allelic variation is decoded, it gets decoded, and it's visible in the form of protein variation. And then we see variation in the species. Is it clear till now? Are you all there, or you are having these black boxes, or can you PlayStation पे हो? So what I have used, and that's the technical term, this is a mutation, this is change in the nucleotide. Change in the nucleotide, which is visible in the form of amino acid change. Amino acid is part of protein, which means it's causing change in function of the protein, and we see that in the form of appearance, or in genetics we call that appearance. In genetics is called phenotype. Soon we will go in detail of these terms. So all the collection of genes जी वारिस
सर मेरा एक क्वेश्चन है आपसे
जी जी
सर ये आपने ये पॉइंट लिखा हुआ है एलीलिक वेरिएशन लीड्स टू हेरेडिटरी वेरिएशन और दिस लीड्स टू वेरिएशन इन स्पीशीज
या
तो आपने तो अभी कहा कि जो प्रोटीन बनती है उसका फंक्शन एक ही होता है बस अमाइनो एसिड लेवल पे डिफरेंस होती है जैसे आपने अभी अपने हेयर की एग्जांपल दी थी तो फिर ये वेरिएशन इन स्पीशीज को कैसे लीड करता है
सो द hair color is different, so that is variation.
जी
ठीक है. Now how this variation arise is due to change in nucleotide sequence, which is resulting in change in amino acid, and this one amino acid change, or maybe few amino acids change, the protein may go into the same cellular function, but its function is compromised. It's not 100% efficient. It may vary. This allelic variation may vary from complete loss of function, all the way to partial loss of function, which means functional protein is there. Is it clear now?
तो सर फिर ये वेरिएशन इन स्पीशीज में कैसे हो गया फंक्शन तो उसका सेम है तो फिर स्पीशीज में वेरिएशन कैसे आ गई
Variation in species in terms of appearance, we said. You remember I gave you appearance kya hai? White hairs hai. And imagine if there was no variation in the allele, will we see white hairs in the population?
सिर्फ फिनोटाइप चेंज है तो स्पीशीज तो होती है उनका डिफरेंट जेनेटिक मेकअप होता है तो वो तो नहीं फिर होना चाहिए
Variation in species means variation in humans or variation in plants. Variation in species, don't read it like humans will start behaving like or will start looking like monkeys.
ठीक है सर.
Phenotype change. Why this phenotype is being changed? This phenotype is, if we don't have allelic variation, will we see white hair phenotype in humans?
No.
So all the humans will have black hairs. It means there will be no variation in humans. Imagine all of us have 100% identical genes. Identical, I'm saying, not similar. Identical means 100% truly identical. Will we see any variation in humans?
No sir.
So variation in humans is gone then, species of humans. Clear now?
जी sir.
Good. So I was telling you, all the collection of genes in a cell is called genome. Genome is the word. And for example, when we are depicting alleles like this, then let's say my brother has homozygous copies of this gene. So here, when we write genes like this, it's called genotype. We just use word phenotype. Phenotype is the appearance. Genotype is your genetic makeup, the allelic makeup you carry. I may have like this, like this. We are soon going to learn in detail genes, genotype, phenotype, why I have written like capital B, why I cannot have like this, why I cannot have like this. We are soon going to delve into these details. So these details, they were discovered, or we owe all these details to Gregor Mendel. Mendel was working in Austria in church. In those times, the science was dominated by church. Okay, and the Botanical Society at that time, they wanted to understand how can we improve the quality of wool. The big question they had in mind was, how can we improve the quality of wool? What are the determinants? Because Mendel was part of Austria, and it was sorry, it was basically that was a time when Austro-Hungarian Empire and the other empire was the Spanish. And Spanish were producing the best quality wool, and they were giving tough time in trade. So the scientific community got together to learn how can we improve the quality of wool. Mendel used a very intelligent approach, which was truly scientific. His experimental system, we'll soon see, was extremely efficient. He incorporated very fundamental, basic math, maybe not math, I would say. And then he incorporated modeling and predictions that, to validate his results. His experimental system was basically pea plants. Okay. And what he did, he said, okay, I have to, before I set up some experiments, try to understand inheritance or try to discover factors which control different traits. Because remember, the big question was the quality of wool. What controls quality of wool in a living organism? And we had no idea in that time. In 1700, 1800, when Mendel was around, and nobody had idea about genes and nobody really knew much about proteins either. So what he did, he used pea plants and he collected their seeds and he grew them. And what he saw, because when you do genetics, remember the first principle to do genetics is your material, the population you are going to use, it should be a pure line or pure population or true breeding. Okay, 3 words I said: pure line, true breeding, or true to type. What all these 3 are synonymous. It means that there is no variation in the population. You grow round seeded plant and it gives you round seeds always. Okay, there's no variation at all. You do this generation after generation. You take wrinkled seeds and they give you wrinkled seeds generation after generation. Already you see he was extremely smart individual. He identified variation because he knew without variation I cannot understand anything. Just like I said, if everybody has black hairs, we will never know that, you know, the hair color or any trait is controlled by genes. And then we knew nothing about genes in Mendel's time. So after being confident that his population is pure, so rounds always produced round and wrinkled always produced wrinkled. So this is now called phenotype. Your appearance, round, wrinkled, black hairs, white hairs, blue eyes, black eyes, green eyes. This is phenotype basically, okay? These traits. So in science, we call this control experiment. You become confident that there is no variation here. You will use them as standards. You will use them as, you will refer to them always in your genetic experiment. Now, what Mendel did, he crossed round-seeded pea plant to wrinkled, and in the progeny, he found all rounds. What he did, he took 2 from progeny. He took now from this progeny, which he named F1, he took 2 round-seeded plants, crossed them, and went to next generation. And what he saw, that wrinkled, they reappeared without any loss. Here, wrinkle was completely absent, and this one was the phenotype. This was completely absent. After this experiment, what he said, okay, round is dominant. So terminologies start coming now. Round is dominant, and the one which was dominated and only came back in F2, he called this trait as recessive. He says wrinkled phenotype is recessive. So remember, dominant and recessive, these are the phenotypes. Okay. He counted them, and what he found, that there were 5,474 rounds and 1,850 wrinkled, and the ratio, when he calculated, it was near 3:1. Okay, 2.96 to 1. It was never precise 3:1, but 2.96 to 1. So what was important that wrinkled, it reappeared without any loss. It was exactly like original parent where he started. Similarly, there was no blending of round and wrinkled, like, semi-round or semi-wrinkled, not at all. They were exactly alike their original parents. So there was no blending of the two traits. And as I told you, the two terms he coined, dominant versus recessive phenotypes. After observing these things, what he did, he said, okay, let's take other traits. And he took purple flowers, crossed to white flowered. He took tall pea plants, crossed them to dwarf pea plants, and yellow colored seeds crossed to green colored seed, and so on. So 7 different traits of pea plant he observed, and he uh, you know um, set up these crosses. And what he discovered, that he always found one trait completely dominant and the other recessive. For example, purple was completely dominant, white was recessive. And again, in F2, this is the F2 population. In F2, the ratio was 3.15 to 1. You take tall versus dwarf, he found same. You take yellow versus green, he found same F2 ratios. So when we say 3:1, so this is called F2 phenotypic ratio. Okay, 3:1. F2 phenotypic ratio. What he did, he said, okay, let's analyze this population, F2 population. He took F2 population and randomly selected 100 seeds, for example, random 100 seeds, and grew them to see what population they are going to produce. What he found, out of those 100, the ratio he found of these phenotypes, so always round, pure round, or true breeding round, pure wrinkled, this one, and the 2 in the ratio he found impure rounds. Okay, why impure? Because they produced 3:1 ratio, 3 rounds, 1 wrinkled. So this gave him a clear indication that 3:1 phenotypic ratio, there is hidden 1:2:1, what he named genotypic ratio. Because there's something different between this round and this round. Although phenotypically they are same, but they are impure. One is producing impure population, the other is producing pure population. What he did next was, he said, okay, let's solve this enigma. What has gone wrong or right in his experiments? What we know, he had phenotypes. He started with phenotypes. What were phenotypes? Round, wrinkled. These were the appearance. F1 phenotype was all rounds. And when he self-crossed the F1 population, he found F2 and phenotype was 3:1 round versus wrinkled. And based on his numerous experiments, he concluded that, you know, these phenotypes, round, they are encoded by a pair of factors. Since round, he gave the dominant trait, he designated that with capital R. Capital R, and he said these factors, he named them factors. These factors, they actually exist in pairs. He said wrinkled, since it's recessive, so it uses small letters. He said from parents to offsprings, each parent only contributes half of the factor. These factors will separate. They will not go in pairs to next population. And the progeny will carry, since each is contributing half, so F1 population will be this one. Now, since I'm going to self-cross F1 round, it means I'm going to have F1 round like this. Each factor is going to contribute, each parent is contributing half of the factor. So its factors are like this or like this. This one like this or like this. How it will look like. So this parent capital R, small r, this the other parent capital R, small r. So it will be like this. Since capital R is dominant, so you have 3 rounds, round, round, and round here, phenotypic ratio, and 1 wrinkle. And if I write them like this, this is 1, 2, 1. Isn't it what we just talked about, genotypic ratio, 1:2:1? Now, once you will grow this one, let's say this seed from this parent from F2, this will give you impure, this will also give you impure, this will give you pure round, and this will give you pure wrinkled. That was the basis of his conclusion. And he said these factors, they exist in pairs. Whenever you work in genetics, you don't just jump on these things, you know. These are genotypes, these symbols. Remember, you will always use capital letters for dominant, recessive letters for small letters for recessive traits. Phenotype, recessive phenotype. And what is happening here? Who would answer? Why each factor is each parent is contributing half? Why not both the factors? Hello? G Ali.
Because if both of them contribute both of the factors, then the number of factors will keep increasing generation by generation. Second generation में 4 हो जाएंगे, फिर 8 हो जाएंगे, और फिर balance नहीं रहेगा.
Yeah, absolutely right. But how it is possible that it contributes half. How this is made possible? What are these? When we are saying they are going half, who would like to answer?
Sir, some students are answering in the chat box.
In the chat box? Nahi,
Yes, sir.
please don't answer in the chat box. I would like to hear from each one of you. Because when I'm giving lecture, I don't look at chat box. You can raise hand and you can interrupt me whenever you want. Feel free. So this is actually meiosis is happening. These are gametes are being formed. Okay. And that's why you are getting half factor coming in. After these numbers, what Mendel did, so why I wanted you to learn about this was, I wanted you to learn that whenever you are going to deal with genetic problems, you are always going to write phenotypes first. Round, wrinkled, tall, dwarf, purple and white, etc. And look at F1 phenotype, then F2 phenotype, and then you work backwards, and you have these genotypes. Then you, after looking at phenotype, you will see which one is dominant, which one is recessive, and then you decide the dominant I will designate with capital letter, recessive with small letters. And also don't make a mistake in life that, what they do, they say, okay, since round and wrinkled are two opposite phenotypes, they give capital R to round, and then to wrinkled they give, let's say, small w or small m or whatever. You have to give, when you are dealing with same trait, seed shape, round, wrinkled, they are same. You have to use same alphabet. R, capital for dominant and small letter of R for wrinkled. This is a very important point. So this is what we just learned. You have, in F2, phenotypic ratio is 3:1, and underlying hidden genotypic ratio is 1:2:1. So far we have used terms, genotype, phenotype, dominant, recessive. New term is coming, which he used was homozygous and heterozygous. This one, for example, a true breeding round, when you have both the alleles are identical, when you carry same alleles, and here same alleles are capital R, for example, this is called homozygous. This is also homozygous, small r. But this one is heterozygous. When two different alleles are present in the cell, in the same organism, two different alleles, that will be called heterozygote or heterozygous state. Okay. So let's take a 5 minutes break. On my cell phone, it's 15:31, and on 15:36 we will start again. So let's start again. So once he found that, repeatedly in 7 different experiments, or 7 different phenotypes, he found that traits are completely dominant, and F2 3:1 phenotypic ratio, as well as underlying 1:2:1 genotypic ratios can be proved easily. He started doing predictions. For example, he said, okay, if I use F1 round, and F1 was round because round was dominant, F1 round, and I cross it with wrinkled, I can very easily predict. He said, okay, if I have F1 round, I know F1 round is like this, wrinkled is, sorry, F1 is not like this. F1 is like this, and so I can predict that I will get 1:1 ratio in the next progeny like this. If I cross F1 tall with dwarf plants, F1 tall will be like this, and dwarfs will be like this, because tall was dominant in his experiments. And he made these predictions repeatedly with different phenotypes, in different experiments, and he proved his results. In genetics, we call whenever we cross F1 heterozygous, whenever you're going to cross it with homozygous recessive parent, that is called, so this thing is called test cross. And remember, ratio of test cross will always be 1:1. Always. Because you are crossing F1 heterozygous with homozygous recessive parent, this will be the ratio, no matter whichever phenotype you take. This is what he did, and he showed repeatedly. Now, Mendel was gone. This is how, when we have these new technologies and beautiful cameras, people have shown that, look, these are the wrinkle seeds. People repeated those experiments, you see, and then these are rounds. 3:1 F2 rounds. And they also characterized the DNA level genes of round and wrinkled. And what they discovered is that actually round is completely normal gene, this one, and wrinkled, it contains a point mutation in the promoter sequence of the gene, and due to that mutation, transcription is not taking place, and there's no messenger RNA being made of wrinkled. So it's a complete loss of function of wrinkled round, and that's why we see this phenotype. Now, it's very important to understand the coding sequence is intact there. ATG all the way to UGA, 100% identical, but since its transcription is not taking place, so this is now wrinkled allele. Allele 2 they call. But there can be more than, many alleles of the wrinkled. For example, if in genetics we call the normal one, the normal allele, or the reference allele with which we always compare, we call that wild type. Okay. In mutant allele, for example, one of this one is wrinkled. Okay. We say this is small r, wrinkle. But this was a promoter, mutation in the promoter. Let's say within the gene here, there is a mutation. And it also can produce wrinkled phenotype. Another place, another nucleotide change, another amino acid change. And so as we see different, or we discover different mutations within the same gene, we call them alleles of the same gene. So A prime, A2 primes, A3 primes, they all are alleles of the same gene. Okay. Alleles are alternate forms of the same gene. Okay.
Sir, I have a question.
Yeah, please.
Do alleles have to have phenotypic change also, or can they just be genotypic?
Very good question. Very good question. So normally, new alleles are identified based on phenotypic variation. Okay. There can be alleles at the genetic level, but at phenotypic level, they can be 100% similar. And there can be, as we will move on in this course, we will learn that some of the mutations, they don't result in change in amino acid. We call them silent mutations. So your question is very good. In genetics, we are mostly dealing with phenotypic variation, and we give preference to when we have a mutation, and then that gives us a specific phenotype. In normal population, for example, in our normal population, there is variation. For example, in the same gene, if you screen, you look at their DNA sequence, you screen, let's say 10,000 people, and out of 10,000 people, 100 or 200 people show one nucleotide or two, or this level variation, but they all look same. They don't show phenotype, no disease, nothing. So we call this polymorphism. Polymorphism means there is variation, but unless we don't see phenotype in genetics, that is kind of not so useful for us. Okay.
Okay, thank you.
So what Mendel did there, you see, I told you he studied 7 different traits. Okay. And he found the dominant one, round was dominant, yellow was dominant, and he also found recessive ones. Okay. And you see, it was never precisely 3:1. It was always close to 3, and he used his intuition. And he rounded it off to 3, and then he used, simple basic math, calculated ratio, and made predictions. Now we call this, when we are dealing with single trait here, round versus ring, I mean the shape of the seed. Sorry. When we are dealing with a single trait, that genetic cross we call monohybrid. Mono means one, single. But after monohybrid, what Mendel did, he went to dihybrid. He said, okay, I know round is dominant over wrinkled and yellow is dominant over green. In monohybrid crosses, what if I combine both? If I say I'm going to use, yellow wrinkled, cross it with green round, are yellow or green going to impact round or wrinkled? Are they going to, disturb each other's inheritance? He knew already round versus wrinkled. We already know 3:1 and 1:2:1 ratio. He also knew monohybrid yellow versus, I just showed you this one from data from, so in monohybrid state he knew. Now, once he combined them, yellow wrinkled, green round, what he discovered, all the F1 was yellow round. And when he, so this sign is, whenever you have this sign, it means self-crossing. He took pollen of the same plant and threw on the stigma of the same plant, okay? He went to F2, and what he found in the F2, there were 9 round yellows, 3 round greens, 3 wrinkled yellows, and 1 wrinkled green. The total numbers were like this: 315, 108, 101, and then these are the ratios. What this is showing you, in a dihybrid cross, when you self-cross heterozygous F1, you will get F2 phenotypic ratio is 9:3:3:1, okay? Phenotypic ratio. But the fundamental question he was asking, are these two different genes? He didn't know the word genes. He used the word factors. He said, is factor that encodes for yellow going to impact inheritance of wrinkled, or green is going to impact inheritance of round when I bring them together? And what he did, he said, okay, let me calculate now. And he combined all the rounds, these rounds plus these rounds. It gave him 423 total. 315 plus 108, 423 rounds. Combine all the wrinkled. So for a moment, forget about now yellow green in the F29331 when you get. For a moment, just focus on one trait, round and wrinkled. Combine all the wrinkled. 101 plus 32. What he found, that the 3:1 ratio is preserved. It's not disturbed. He did same thing with yellow green. So this yellow plus this yellow, 315 plus 101, 416. Recessive for yellow was green, 108 plus 32, 150. So the conclusion was that underneath 9331, there are two hidden 3:1 ratios, but the more important conclusion out of dihybrid cross was that, when you deal with two different factors, none of the factor disturb others' inheritance, and they assort independently when they go from parents to offsprings, okay? And in F1, these parents to offsprings, whenever you have, dealing with two different factors, and we can today in 21st century, we can use the word genes because they turned out to be genes. Two different genes, they will always inherit independent of each other during meiosis. They don't disturb each other. And that was a second law of Mendel. The first law was this one, which he discovered in the monohybrid cross, that the two factors or alleles of two genes, they can be same alleles, they can be different alleles, they can be same alleles, or they can be different alleles in the same cell. They will always segregate, and here the word is segregate, because the two in meiosis, this is meiosis is happening. The two are not, both the alleles are not going to end up in the same gamete. Okay.
So what if the genes of the wrinkled and of the colored were on the same chromosome?
Please repeat your question.
Sir, what if both of the genes that he was studying were on the same chromosome?
What if, yeah, hold on. We will go there when they're on the same chromosome. There are many people in this class who haven't taken Bio 101. So that's why I'm going a bit slow. So we will talk about recombination and linkage, etc. But for Mendel never observed linkage or recombination. That's why he concluded that, you know, the two alleles of the same gene or same factor, they segregate independently, and they segregate from parents to offsprings. Yeah.
But if he had been studying linked genes, so how would he come to a conclusion of factors?
His conclusion would have been messed up. He was very lucky. He was very lucky because all these 7 traits which he studied, all these 7, in 21st century now we know, we have the luxury to know, all these, so P's, they have 7 chromosomes. And genes for all these 7 traits, they are on independent chromosomes. He was that lucky. He was extremely lucky. So if linkage or recombination came across his experiments, I think Mendel would have been, you know, in big trouble. We will talk about this all. So now, what I want all of you, what I want all of you is to pay a little attention and learn something which is very important. You are dealing with dihybrid cross. Sorry, why it goes itself. So why it's not working? My pen is not working. What went wrong? Just a minute. Let me change the thing. So something went wrong with my pen. Najma, can you come to my office, please? I need to change. My hand is broken, so I cannot use both hands. Najma, ये जरा निकाल सकती है ये? हाँ, देखो ले पाती है यार, मेरा ख्याल है वो निभी आती है सिर्फ इसकी। क्या हुआ है? वो यूज मैंने इसे काफी यूज किया है ना, तो वो हो जाती है फिर then you need to change new यार हाँ इसे रोटेट करके देखा तो ऐसे यही से होगा यहाँ से मेरे हाथ अगर होता नहीं तो भी कंटिन्यू विद द क्लास
कोई और यूज करते हैं?
let me finish class and then I will. Okay, so what you should do is that whenever you are dealing with dihybrid cross, you should never use there are two methodologies. One is called the Punnett Square, which is here. What we have done, we have, you see, this one, the gamete formation, you need to make gametes. So capital Y, when we say they always inherit or assort independently, it means Y, capital Y can go with capital R, capital Y with small r, small y with capital R, small y with small r, which are written here. 4 different types of gametes which can be formed. And since you are having this other parent itself crossing of F1, you will have the same. And then if this fertilizes this, you will get capital Y, small r, or this one, capital Y, capital R, small r. It takes you long to solve a genetic problem. And at undergrad level, this is not allowed in my class. I would like you to learn the branch diagram. And branch diagram is very simple. It's fun, I tell you. It's so fast. Okay, good. Thank you. So it's हो गया 1 मिनट में ये लगा है. नहीं इसका ना कोई मैंने नहीं इसमें भी वही मसला आ रहा है. फिर वो निब का मसला नहीं है. There is मैंने ना कोई इसका वो इसे दूसरी को लगा दें वो ना आई थिंक ये बटन कोई प्रेस हो गया जिसकी वजह से या वो फॉरवर्ड हो गया. हालांकि नॉर्मली वो फॉरवर्ड नहीं होता. थैंक यू. I'll figure it out after the lecture. So instead of spending a lot of time this way, what we do, we draw branch diagram. And to understand branch diagram, what you do, you normally use, when you are having dihybrid cross, let's say you have capital Y, like which was here, capital Y, small y, capital R, small r. You focus on single trait first. For example, we will say capital R. If we have capital Y and small r in a monohybrid cross in F1 and we go to F2, how many will be with capital R? I repeat my question. I know it's online, so it's difficult to understand. If we consider at the moment only round and wrinkled, and we have heterozygous F1, which means capital R, small r, and we are going to F2, how many in the F2 individuals or progeny will have capital R?
3.
Yes, very good. Easy, because you have 3:1 ratio. So what you do, you have written here 3/4 in F2 round and 1/4 wrinkled. Clear? Now you have this one. Now we will draw a branch. Why? Because we know we have this is the parent whose gametes we are making, and we want to go further to F2. Now once we have decided 3/4 round and 1/4 wrinkled, now we will pay attention on this thing exactly similar way, but we will make branch. Look at the direction of the arrow I have. The branch will be from this to this an arrow, and the other arrow will be from here to here. Why? Because 3/4 will be yellow. Now we are only focusing on the yellow, heterozygous yellow, and 1/4 will be greens. Similarly, we'll have, if there is wrinkled here, and we say they assort independently, here 3/4 will be yellow and the other arrow will be 1/4 green. And to find the numbers, phenotypic numbers, what we will do, we'll say 3/4 capital R dash. We say dash because it can be capital R, it can be small capital R, small r as well. So 3/4 will be capital R multiplied by 3/4 of capital Y. Okay, it will give you 9/16 for capital R, capital Y. Now the second branch, 3/4 of capital R, this one, multiplied by 1/4 of small y, and you will get 3/16 capital R, small y. Let's solve the other part, which is the 1/4 of small r, 1/4 of small r, multiplied by 3/4 of capital Y, multiplied by 3/4 of capital Y, and we have 3/16 small r, capital Y. And 1/4 multiplied by 1/4 of green will get small r, small y. It brings you to these numbers we just saw. 9331. Sorry. So can the 9331 ratio, can this 9331 ratio be explained simply at the gametic genotype? Yes, we are actually looking at the gametes here. When we say, you know, capital R small, capital Y or capital R small y, here normally, and when we are looking at phenotypes, what we do, we can simply have dash here, capital R dash capital Y dash, because in phenotypes, phenotype is being determined by the dominant allele, capital R and capital Y. The other allele can be small r, other allele can be capital R. Same for small y or capital Y. Okay. Now you can also make gametes. Gametes are even more simple. You have F1 heterozygous. F1 heterozygous was this one, capital Y, heterozygous for Y and heterozygous for round. Let's make their gametes. Half of the gametes will be R, okay, and half of the gametes will be small r. Now branch, half of the gametes will be of these R will be capital Y, and half of the gametes will be small y. Because remember, the two alleles, they never go to the same gamete. And we saw already here, either it will be half this one and half this one. And similarly, when they assort independently, when genes are on independent chromosomes, R can end up with capital Y or R can end up with small y. Small r can end up with, you know, capital Y, or small r can end up with small y. Half of these small r. This is a typo, not capital R. So I would like you to, today they will upload an assignment for monohybrid and dihybrid cross. And I would like you to learn branch diagrams, because in next lecture, what we are going to do, we are going to actually come to more complicated picture, which is what one of your colleague was pointing out, that what if they are on the same chromosome? So Mendel saw this situation where, you know, round or wrinkled allele, so the shape, seed shape gene was on one chromosome, and color, seed color gene was on totally different chromosome, independent chromosome. And Mendel found these, you know, when he goes through these gametes, you see, we made 4 gametes possibility, and they are like 25% each, 1/4 each. So Mendel found these ones, but when Mendel was gone, and in Mendel's lifetime, nobody really took him serious or his discoveries. It was only in 1804 or 5, I believe, when they rediscovered Mendel. They were studying meiosis, and while studying meiosis, they found that you know, some of the cells in fruit fly, they contain half the number of chromosomes as compared to majority of the cell types in the fly body. And the cell types which contain half the number of chromosomes, they are actually the gametes. So here, for example, this one contains how many chromosomes? 1, 2, 3, 4. Okay. And gametes contain 2, half the number of chromosomes. So these ones are called diploid, and they are represented by 2n, small n, and we call this ploidy. So in normal cell types in our body, the chromosome numbers are, like, for example, in humans, they're 46. But in gametes, in sperm and oocytes, it's 23. And that is a result of meiosis. So your gametes are haploids as compared to somatic cells, which are diploid. So after rediscovery of Mendel, and, you know, once they learned that, you know, these that meiosis is actually resulting in segregation of the alleles to opposite to different gametes. And they also discovered that, you know, genes can be on the same chromosome. Not necessarily they should always be on the same on different chromosomes. So what if genes are on the same chromosome? One of your colleague was asking question, what could have happened if round and yellow genes were on the same chromosome? Will the results of Mendel be still like this, 25% each gamete and 9:3:1 ratios, or we should expect changes in what Mendel discovered. So this is what we will talk in next lecture, but I would like you to do the monohybrid and dihybrid questions. Few of the questions they will upload, Najma and Abdullah. These are the TAs for the course. They will upload these lectures, assignments you should do, but I would encourage you to go and do as many practice questions as possible, because your first exam is going to be after 5 lectures. And since this outline clearly indicates that you have 4 exams, and it makes this course easy as well, because you take 5 exams, and then next exam will be, I think, after 6 lectures of after first exam, and that's how you learn genetics. But you will learn genetics if you do more problem science. And today is a start, so I would encourage each one of you to do as many practice questions as possible for each lecture when we will upload them, but go and they are freely available all over, so that you know when you are solving a genetic problem, which gamete, how to give symbols, capital R, capital A, small a, heterozygous, homozygous, diploid, haploid. The more you practice, the easier it will be for you to learn the initial 5 lectures, which are really interesting. They're really fun if you really get involved in these solving practice questions. I think our time is up. We have seven more minutes. If there are some questions, please ask. Feel free to ask questions. Ji Arshay? Are there some questions?
Yes, sir.
Ji Adan.
So we know that genes control traits. So if there is a variation in genotype, so will it affect its phenotype?
If there is what? Sorry?
If there is a variation in genotype.
Yeah, if there's variation in genotype,
So
yes.
that variation, so can that variation affect the phenotype?
Of course, it depends on the frequency of variation. As we said, there could be some variation which is silent, which does not result in change in amino acids. Okay, so we may not see any change in protein function and no variation. But if there is variation which is in the genotype, which translates into protein or absence of protein, we will clearly see variation.
Sir, but is it necessary कि अगर phenotype में अगर genotype में variations हैं तो उसकी phenotype में लाजमी होंगी?
If so, the question is it necessary if there is variation in the genotype, so you will see variation in phenotype? No.
Yeah. Sir, but genes control traits. So
Yeah.
traits तो phenotypes भी होती है मतलब.
So phenotype is general appearance, and then if you go and for example, height is a character, eye color is a character, and then within a character you have small, dwarf, and tall. These are traits. Blue colored eyes, black colored eyes, green colored eyes. These are traits as well. Am I making my point clear? Are you getting answer to your question?
Yeah.
G. Other questions?
Sir, I have one.
Yeah.
So mostly we see green peas here in Pakistan. So
Yeah.
can we say that the yellow dominant trait has been eliminated and the recessive green color has been selected by natural selection here?
Yeah. Not natural selection, I would say. So people preferred the green peas, so everybody grows green peas. For example, for a long time, you might have seen the कैप्सिकम, ये जैसे वो बड़ी मिर्च नहीं होती, हम लोग उसे उर्दू में शिमला पहाड़ी मिर्च कहा करते थे, पता नहीं आपकी जनरेशन कहती है कि नहीं कहती. तो for long time we only saw green colored capsicum, but now we have yellow, we have red, so people selected their bread and you know, they are being used as well. So in Pakistan we only see green because people like it green, or all over the world maybe they like green, but it's not that the nature selected out against yellow.
So we can say we artificially selected this.
Yeah, artificially we preferred, we selected artificially. Yeah.
Got it.
Genetics is all about, you know, selection, natural selection as well as artificial selection. Any other questions? So let me also tell you now that all the questions in your first exam, they will be genetic problems, monohybrid, or, you know, as we move on, you will see today you saw a lecture. This is just a refresher lecture. The real things will start from next lecture, I would say, and then next lecture you will see. And if you do more practice, you can score very easily 100%. Let me also tell you my own experience of this class. Anyone who scored an A grade in this, and by mentioning this, it does not mean you should go and try to get A. I strongly discourage this grade consciousness. But anybody who scored A, and that means above 80% marks, I haven't seen them, you know, failing any other exam, because this exam is that this not exam, this course is that analytical. It's very interesting course. No big maths, rather I think there's no math, maybe only slightly we will talk about Hardy Weinberg in last quarter. Very briefly we'll touch upon that in the population genetics, but it requires analytical approach, very analytical, and I think you will enjoy that. Historically, I had many computer science people in this course. All the biologists who took this course, I think they all, after 3 years, I didn't see them in Pakistan. So they were all mashallah. So pay attention to every course what you study, but this biology course is very different. I'm giving you a caution. It's not usual biology course. It will require you to think with a reason more than what we do in other courses, maybe where we have cloning experiments, where we have, you know, protein purification experiments, where we have, you know, experiments to fish out one protein with another. Here you will learn, you know, everything gene based, and then you can score, and trust me, very easily you can score 100%. 80% is no big deal if you are a hardworking student and you regularly do your assignments and do a lot of practice questions. ठीक है जी, it's time. I wish you all good luck, and I look forward to a wonderful semester with all of you. We have to decide with your TAs time for recitation. Each week there will be one recitation, okay? And in the recitation, they will help you with the, or in their office hours, they will help you with the problem sets, these genetic problems, okay? So I'll send you an email and we can decide where you have free slot, time slot, so that we can schedule your recitation on that particular time slot. I'll send you email today with some options, or if I see you cannot come down to single time slot, we may have to do then Google poll kind of thing, so that you know, it will be good if we start right from this week, so that you know 5-6 lectures के बाद एग्जाम आ जाना है, ठीक है? तो बड़ा important है you solve as many genetic problems. Okay, Allah Hafiz.